Raviteja Venchirla, Assistant Professor, Department of Pharmacy Practice, MNR College of Pharmacy, hosting the day Conference 2K25. The one-day national conference on advances in drug discovery, development, and patenting challenging and opportunities aims to bring together researchers, practitioners, and policymakers in the field of pharmaceutical science to explore the latest advancements and challenges in drug discovery, development, and patenting. The conference will provide a unique platform for sharing knowledge, professional growth, fostering collaboration, and addressing issues within the pharmaceutical industry.
The pharmaceutical industry plays a crucial role in improving global health care. It requires integration of scientific research, clinical testing, regulatory approvals, and intellectual property management. This conference will offer valuable insights into the multidisciplinary approach needed for the successful drug development and patenting, focusing on both opportunities and challenges. The MNR College of Pharmacy is located at the MNR Herald campus at Sangareddy. It was established under the MNR Educational Trust in the year 2004. And since then, it has been nourishing the pharmaceutical profession. It is one among the 78 renowned institutions of the MNR Educational Trust. MNR College of Pharmacy is approved by the Pharmacy Council of India, a statutory body of the Government of India. The college also has been accredited by NAC and is affiliated to Osmania University, Hyderabad, for all its courses which include B.Pharm, M.Pharm in Pharmaceutical Chemistry, Pharmacology and Pharmaceutics, PharmD and PharmD Postbaccalaureate.
Along with academic excellence, MNR College of Pharmacy excels in extracurricular activities, which includes sports and cultural competitions, entrepreneur and communication skill development programs and competitions, careers and competitive exam guidance, as well as seminars in research and development. The institution encourages social activities through its National Service Scheme and Institution Innovation Council for the holistic development of students.
Now we have the first session of the day. I request Dr. Vishaka Kulkarni, Professor and Head of the Department of Pharmacognosy at MNR College of Pharmacy, to acquaint the speaker of this first session to us. Thank you, ma'am. Good morning and welcome to One Day National Conference. Today, I am delighted to introduce our first resource person for the scientific session 1, Dr. Shweta Mahajan Kulkarni, who is distinguished personality in the field of patenting. Shweta Mahajan Kulkarni has completed working as an associate director at IPDO Dr. Reddy's Laboratory in intellectual property management from last 18 years. Madam has graduated from Delhi University with a gold medalist in 2000 and it completed her post-graduation in Delhi University with a second rank holder. She has completed her PhD in Biomedical Research Center in Delhi University and diploma in intellectual property rights. Madam has more than 15 publications in national and international journal. She has qualified GATE entrance 2002 and CSR Junior Research and Senior Research Fellowship from 2002 to 2006. To her credit, she has awarded with the Sheshadri Award from University of Delhi for being a first rank holder in the BSc. She has received the Dr. Ashish Ganguly Fellowship. So Madam has a credential of many fellowships as well as the experience and expertise in the patenting. We, on behalf of MNR College of Pharmacy and MNR Educational Trust, MNR University, welcome you, ma'am. Please come on the dais and take a charge of the session. Thank you, ma'am. Thank you for joining us.
No, that's fine. Pointer? morning, everybody. It's already. So first of all, I would like to convey my sincere thanks to the principal sir for inviting me and giving me an opportunity to interact with all of the young minds here. So thanks a lot, sir. And thank you to Dr. Vishakha also, because she was coordinating with me from last, I would say, 1 week and adjusting with my schedules. So without wasting any more time, I think we can start with the first session, which talks about role of intellectual property rights in pharmaceutical industry. So what I understood from Dr. Vishakha is few of you might be having some basic understanding about the intellectual property. What does it mean? So it's not working. Oh, okay. Maybe, maybe they can just keep on scrolling the slides. Sorry? You can just keep on scrolling the slides. Okay. So let us start with the basic definition about what is intellectual property. So as the name suggests, intellectual property is nothing but the creation of human minds. And for a creation which is having novelty, which is having industrial application, various countries give rights for those creations in return of the fact that whosoever is creating that invention, they will be disclosing certain aspects of it to the public at large. So this is what is called as intellectual property. However, there is a lot of debate going on about the artificial intelligence also. You might be knowing, much better than me, because you could
Be using ChatGPT and whatnot for completing your projects, right? So we have seen that AI is one upcoming tool which can also lead to the creation of, which could also lead to the creation of inventions. And now various countries are debating about it. Shall it be given a right for patenting or not? So a few regulated markets like US, Europe, they do acknowledge the inventions which are created by AI, and they are in the process of giving the rights for such inventions
that are created by machines. Moving forward, what are the different types of intellectual property? So intellectual property is a big umbrella. Under this, there are multiple rights, which a company, which an inventor can protect. For example, copyright we see in the form of books, in the form of journal articles, in the form of any label on any packaging. All those rights are copyrights. Nobody else can copy that, for their own products, for their own articles. Second is trademark. We all are aware of the symbols, the marks, the words. The well-known example is Coca-Cola, right? The way Coca-Cola is written, only that company can utilize this trademark for their products. No other company can utilize it. So there is a legal right which is given for the trademarks. Trademarks have to be registered in the agencies, and a certain tenure or protection will be given to the trademark. Likewise, there is also geographical indications. We all are aware of the Tirupati Laddu, right? So it belongs to the specific region of Tirupati. And if it is made at any other place, it can't be called or it can't be given that GI. So GI are very specific to the region from where they belong. Another example is Alfonso. Alfonso, it is a variety of mango which is allocated for the Konkan region. So any other mango which has grown outside the Konkan region can't be tagged as Alfonso. So again, for geographical indications, there would be certain marks, there could be certain terminologies which will be given, and that is given by the agency, by the government agency. Coming to the fourth aspect, it is the trade secret. This is also one form of intellectual property. Now, as the name suggests, it is a secret information. It will not be in public domain. There will always be certain nuances of the invention which a company or inventor do not want to disclose to the public at large. Now take the example of Coca-Cola, right? We all know it is a drink, it is black in color, it is sweet, but we do not know how it is made. What are the ingredients of it? So the secret formula which that company is using to make it, that is a trade secret in this case. It is not a very commonly followed practice because any company will have to, make a balance what to disclose and what not to disclose. And especially in certain industries, the things are very porous. It is very difficult to, keep secret information intact within the organization. So trade secret is a choice which has to be made very cautiously, and not every company will follow that practice. The fifth one is the most important one, which is the patent. Now, patent is given for the inventions. Unlike copyright, trademark, GI, trade secret, in case of patents, you have to invent something. You have to disclose it to the public. You have to show what advantages it has over the things which are already available in the public domain. Unless all these things are shown, agency will not grant a patent or to the. Okay, so taking the case of intellectual property right in pharmaceutical sector, the example of Zovirax. You might be knowing that this is a topical cream which is consisting of acyclovir. Acyclovir is a very known drug which is used to treat the symptoms of viral infection. Now, in this particular case, what are the different ways for a company to protect the intellectual property? One is definitely patent, which means that acyclovir as a product, as a compound, when it was invented for the first time, it was protected by virtue of the patent. And it happened somewhere in 1977, because that patent got expired in 1997. Then there is another intellectual property in this case, which is the trademark. Now, Zovirax, which is the name given to this product, which is by GSK, only GSK as a company can use Zovirax as a trademark. If any other generic company, be it Dr. Reddy's, be it Mylan, anyone wants to use this brand name, they can't use it because it is a specific to GSK. Now, the fourth one is design. Medicine, we will be getting a packaging information leaflet. That is nothing but the copyright. And if any company to use that information, they can't just go and copy it and place it in their own product. No, that is not allowed. Next slide, please. Okay. So amongst all the intellectual property which we have, just had a glance of it, the most important one in the case of pharmaceutical sector is the patent. It's not that trademark, design, or the aspects are not important, but the maximum, I would say, the driving factor are innovating the drugs or the companies who are copying the drugs. For both of them, patents is the most important aspect. Now, what is patent? Patent is, again, as we discussed about the other aspects of IP, patent is also a legal right which is given to an applicant or to an inventor in return of the public disclosure of their invention. Now, what does it do? Why do I file patent? The only advantage which a patentee would get is they can avoid or they can prevent others from copying it. Now, again, the relevance, becomes a question mark. If I invent something, if I file a patent, does it mean that automatically I will be getting money out of it? The answer is no. Once you file a patent, you also have to see what is the monetary benefit, how it is beneficial to the society at large. Whatever are the existing therapies in public, is it behaving better than the existing therapies? If the answer is yes, you have a couple of options. You can either continue with the development of that compound, till the approval of that as a drug from the regulatory agency, which is an expensive affair, and it takes a lot of time. It takes almost 10 to 12 years to perform all the clinical trials, etc. A lot of money is involved, which every company or every institute can't afford. Then why institute should file patent? Now, what is the incentive for them to do inventions and file patent? The other incentive would be, if you file a patent for a promising compound and you have, enough data to prove that it is beneficial over the existing compounds in the art, you can attract the investors, you can attract the big innovator companies. They can buy your patent portfolio along with the drug, and they themselves can develop this product because they have the muscle power to do it, which every institute or every small organization may not. Even the small institutes or small companies prefer to file a patent application or get a grant of the patent to secure the legal right, because you never know when that drug may create a monetary benefit for you. Last but not the least, whenever an institute or a small company is filing patent applications, there are certain provisions, which are given by the government, which helps in the tax exemption. So this is also one of the reasons by which companies or institutes file patent applications, definitely for the things which are novel and which are inventive. So these are the reasons why whenever we are inventing, we are developing, be it an incremental invention, we should give it a thought. Is it something which can be patented? If the answer is yes, we should go ahead and file it. Now, amongst patents, again, there are three different categories of patents. One is design, second one is utility, and third one is plant. Design patents are granted for the aesthetic part of it. Whatever invention you do, if it is related to packaging, if it is related to certain device, it will come in the design category. Now, utility patents are basically for the products. So if you invent a new chemical entity, or if you invent a novel process, a very cost-effective process for already existing compound, that will come under the category of utility patents. Third is the plant variety, which is mainly for the new varieties of the plants.
For that also patents are granted under the plant category. Now each kind of patents have a certain tenure associated with it. It's not that once you file, once you get a grant of the patent, it's for unlimited life. No, it's not like that. Though you are disclosing your details, agency give you right, the legal right, only for a certain tenure. In case of design patents, it will be only for 15 years from the date of file. The moment you decide you have to file a patent, from that date, it will be 15 years. In case of utility inventions, it will be 20 years. In case of plant, again, it will be 20 years. Now, a question may come, is it everything is patentable? The answer is no. There are certain things, discoveries, if you discover something which was already existing in the nature, you can't get a patent out of it. Similarly, abstract ideas without having a proof of conception, without having a tangible product in hand, you can't get a patent out of it. So there are certain, I would say, disclaimers which cannot lead to a grant of the patent, and everyone has to keep a watch about it. Henceforth, we will be mostly talking about the utility patents because that is the most important kind of patents which are of relevance in the pharmaceutical industry. Can we move to the next slide, please? Now, there is misalignment. So the question or thought may come, what does patent constitute? How does it look like? We all know about the books, we all know about the journal articles. How do they look? But I'm not sure how many of you have actually seen the patent. So patent is actually having the main 7 components. First one will be the bibliographic data, wherein the names of inventors would appear, the name of organization or the institute on behalf of which inventors are filing the patent application. The filing date of the patent application, in which category of chemical class it is falling, all those basic information will come in bibliographic data. Second would be the abstract, wherein you briefly describe what invention is all about. Then the background, then the background, wherein you will be explaining, suppose you have invented compound A. Now, is there anything in the literature which is close to compound A that already existed in the art before you invented it? If the answer is yes, you will be disclosing about those compounds in the background and you will try to prove the superiority of your invention over what has been existed. Then comes the summary part of it, wherein again you briefly define what your invention is all about and what you are intending to claim. I will come to the claim part
because that is the most important part of the patent. Fifth one is drawings and figures. If your invention need any kind of drawings, if there is a, obviously a chemical compound would need a drawing. You need to represent the structure of the chemical compound. Similarly, in case of inventions which are related to crystal forms, you need to disclose what is the PXRD pattern of it. In case of inventions related to device, you need to disclose how the device looks like. So depending on the need, this section of drawings and figures have to be filled in. The sixth part is the detailed description, which more or less define again the same thing, wherein you are explaining your invention in detail. You tell what is the most promising region where your invention works better or the best. Last, but the most important part of the patent would be claims. Everything you cover in background, detailed description, summary is important, but the most important is what you are covering in your claims. Because the legal right which is given to your patent is entirely dependent on what you write in your claims. Giving you
Give you an example.
if you come up with 10 compounds which are absolutely novel, nowhere reported in the literature, you decide to file a patent application for it. You disclose all the 10 compounds in your detailed description. Over a period of time, when you study more about it, you realize that not all 10 compounds are, biologically promising. It's only
5 compounds which are biologically promising, and then you decide, let me claim only 5 compounds. Let's not claim all the 10 compounds. The legal right which will be given to your patent will confine only to those 5 compounds, not to the rest of the compounds. So you have to be very cautious what you write in the claims. Even if something is not usable for you at this point of time, but don't hesitate to claim it. You never know, the compounds which you left out may become promising in other areas, and then you lose your right if you are not claiming it. So these are some examples of how, patents look like. Just one point, patents are territorial in nature, which means that once you file a patent, once you get a grant of the patent, it is not that it will be globally applicable. No. If you want to have a legal right in India, you have to file in India. If you want to have a legal right in US, you have to file in US. Similarly, in Europe, Canada, in whichever countries you want to have legal right, you have to go file patent application, get it prosecuted. So this is an example of a US patent. This is a front page which mainly discloses the bibliographic data, wherein you can see, at serial number 54, you see the title of the invention. Then at serial number 45, it is the grant date when this patent was granted. At serial number 10, it is the patent number, which is the granted patent number. So I will come to it. What do we mean by granted patent and the application? So this is how the US patent front page will look like. And you also have the abstract in it, which will be just giving you a glance of the invention. Can you move? So likewise, we have Europe patent and the India patent. So this is the face of the granted Indian patent. It looks simple. So this is just disclosing the granted patent number, the title of the invention, and the date of filing when it was for the first time filed, and the corresponding application number. Now, so there is an application number and a granted patent number, which means that once you file any patent application in the patent office, first an application number will be allotted to it. It has to pass through certain stages of examination. You have to convince examiner of the various patent offices that whatever you file is novel, is not known in the literature. It is having inventive step. That means whatever close compounds exist in the nature, your compound is better, is beneficial over the existing compounds. And your compound is going to have an industrial application. So there are certain criteria which each patent office follows, and these are the basic criteria which US, Europe and India follows. Once a patent applicant satisfies under all these criteria, then a patent will be granted, and it will be given a granted number, which you see at the top, which is starting with 210. And the second row is application number, which means that when you file, agency automatically gives you, assigns the application number. Now second is talking about the application number. If you don't take it to grant, you will not get any legal rights. So any patent application which is in public domain, not taken till grant, will not have any legal enforcement rights. So in pharmaceutical industry, there are various categories of utility patents, which, whether it is a generic pharmaceutical or an innovative pharmaceutical company, they try to protect their inventions. First and the foremost is the new chemical entities. So definitely if a company is coming up with a novel compound, which is best in class, which is going to treat a very critical illness, they are going to first claim or file a patent for that compound as a molecule per se. Whether it eventually passes the clinical trials or not, that is a secondary thing. But as I said, you have to first secure the filing date of it. So all the pharmaceutical companies who are into the area of NCEs or even the institutes, I would say, whenever they see a novel compound which based on their prima facie studies, it could be in vitro dissolution or any parameter which you study, you see them as a promising compound, you should go ahead and file a patent application that will fall in the category of NCEs. As companies work further on the NCEs, they may come across secondary patents, which means that they try to further improvise on their invention. One category is salts. So for example, if a free base is not soluble, it is efficient, it is efficacious, but it is not getting solubilized, then how a formulator will formulate it? So practically it is having no utility. In such cases, companies try to develop different sorts of it, different polymorphs of it to make it better and better. So these are the various categories in which any company will be working on it, and they file patents falling into these categories. So we will see how the claim look like for each category of the patents. Yeah, so this is the claim of a NCE category, wherein it was claimed like a Markush structure. So in a layman's language, Markush structure would mean a general formula, wherein you have not defined what your R2 is, what your R3 is, what your R4 is. You just know that this bicyclic system, having a nitrogen at so-and-so position, it works well in certain category of receptors. At this point of time, you are aware of only this point. You will try to protect as many compounds falling in this category as possible. This claim will cover millions of compounds. Even if this company or this institute has not invented, has not tangibly seen all the millions of compounds, they will try to claim it. And they can get claim for it also. What they have to do is, they have to show at least 50 compounds they have made, at least 100 compounds they have made, and they have shown the efficacy of it. That much is fine. So this is a Markush claim NCE example. Now we will see how the specific claim look like. Now this is a very specific product in which a claim has been granted for single compound. So for this company, out of millions of compounds, this is the compound which is finally efficacious, which went up to clinical trials, and therefore they have taken a grant of this patent separately. It's not that they have taken a grant of this patent only, it is a separate patent. Yeah, we will move to the next slide. Now these are the salt claims. As I said, sometimes when a compound is invented, you know it is efficacious, you went on the clinical trials also. In certain situations, during the clinical trials, you experience that there are certain issues with the free base or the free amine. In that case, you try to make a salt of it. And you can do that even at phase 2 or phase 1. So when companies do that, they file a patent for the salt. So these are the examples of the salt claim. The second example is for a blockbuster drug which probably you might be knowing. It is sitagliptin phosphate. It is used for the treatment of diabetes. And this is by Merck. This is an example of polymorph claim claim, again, within the same salt, it can exist as a crystalline form, it can exist as amorphous, it can exist as co-crystals. Not all of them will be usable. Whichever is best usable form for an innovator company, they will try to protect the claim for that. And that's what we have seen in this case. These are the examples in which the crystalline polymorph of the salts or of the free base have been claimed by the innovator companies. Can we move on? This is the example of a process claim. So this compound can already be known in the art, and you just come up with the best process. Whatever process is reported in the art, that might be very expensive, that might not be a greener process. You decide and you invented a process which is very cheap, which is very green, and therefore you decide to file a patent application for it, which is of very use. Why? Because innovator companies tend to go for greener and the cheaper processes. So even if innovator has launched their product, but if you come up with a process which is green, which is cheap, they might come to you and take the license of your patent. Can we move? These are the pharmaceutical formulation, which means that once you combine your active ingredient with the excipients, what kind of excipients you want to use? Is it an immediate release? It is a delayed release? Is it oral suspension? What kind of formulation? Depending on that, companies will be securing patent for the formulations. Yeah, please move on. This is combination with other drugs. In certain cases, a single active ingredient may not be as efficacious as it is in combination with any other active ingredient. In such cases, companies tend to file patent and get a grant of it for the combination therapies, for combination products. So this is an example of a combination patent wherein you have a combination of two different active ingredients, and you see that individually active ingredient is not efficacious, but when you combine them, it has a synergistic effect, and therefore it becomes a more promising candidate for clinical trials than an individual active ingredient. Yeah. This is method of use or a regimen. This is quite heavy, I believe, for you guys. But in a layman's language, what I can say is, if you invent a new chemical entity and you see that it is having an activity for type 2 diabetes or it is anti-cancer, you will claim the use of that active ingredient for treating certain diseases, and that is what we call as method of use claims or method of use patents. Yeah, please move on. This is device. So in case of auto-injector products, you will see the patents on the device part. Certain times it can be, a single dose device. Certain times it can be a multi-dose device. So depending on the nitty-gritties of it, companies protect the device components, and you get a tenure of around 20 years even for device. It is different from the design part. Yeah, move on. So far what we have seen is there are different kinds of patents which are of relevance for pharmaceutical sector, but they do not come all together, So there will be lag when they are coming, and it comes usually in a staggered manner, which we routinely call them as a first generation, second generation, and a third generation patents. So what do we mean by first generation? The moment you see active ingredient which is new, you go and file. That is the first generation patent. Second generation is when you are working more on that active ingredient, you realize that salt is beneficial, polymorph is beneficial. This process is better than what you followed to make the first generation patent. So all these improvisations or incremental innovations will fall in the category of second generation. In the case of third generation, so big companies, innovator companies, they tend to do the life cycle management of the patents and of the drugs. So this is when the third generation patents become important, when they realize that my product is behaving good for type 2 diabetes, but what if I make an extended release formulation of it? Will it behave better than the IR formulation? Will it cater the pediatric population if I make an oral solution of it? Likewise, during the life cycle management, we will see the third generation patents also. Please move on to the next slide. So the key features of the patent, so far, whatever we have studied, these are the important points which we should take away with us. First is, there is a limited duration of patent. Patent is only territorial. Wherever you file, you will have a territorial right. For India patent, your rights are limited only to India. For US, your rights are limited only to US. Enforcement of patent right. You can enforce your patent only once it is granted. In the application stage, during the prosecution, you cannot have the legal right, you cannot enforce it. There are certain nuances, there are certain examining criteria by virtue of which the patent offices would be granting the patent. Now the important ones are, it has to be novel, it has to have industrial application, it should be inventive. Last but not the least, yes, there is a way to challenge patent. It's not the end of the story. I mean, once you get a patent, it's not that you cannot use it for entire 20 years. If you think that patent is not novel, that patent was not inventive, you can go and challenge that patent with the credible grounds in the courts, and then patent can be revoked also, provided you convince the judge. Next one. So this slide is depicting just a snapshot of where patent intersects with the development of the drug. So as we discussed, suppose now you invent a compound, you go and file a patent application for it. That is your year zero.
Once you file it, if the patent is granted, you will get 20 years from the filing date, which means from today you will get 20 years of legal rights. Now, today you are not clear whether this compound will get approved as a drug or not. You will be studying the clinical trials, etc., which will take 10 to 12 years for the drug to get approved. So essentially, out of 20 years of term, you have lost your 10 years while you study the clinical pathway of it, getting approval of it. Again, 1 to 2 years it takes for the approval of the drug. So if you see 10 to 12 years for clinical trials, 1 to 2 years for the approval, essentially out of 20 years of your patent term, you have lost 14 years. You cannot make any money out of it. In order to recoup the investment which big innovator companies do or any company do, there are certain provisions which has come up by few countries, like we see in US, in Europe, in Japan also. The patent offices grants patent term extension, which means that initially the patent was about to expire in 20 years, but if there is any drug, there is any pharmaceutical compound which eventually got approved for the treatment of human beings, that patent can get a further extension of 5 years. Only in such cases, there is a possibility that your patent tenure, your legal rights tenure can be for 25 years. It will not be for all kind of patents. It will be only for those patents which have gone through the clinical trial, which have gone through the approval cycle, because of which they cannot monetize their patents. Yeah? So again, this is a snapshot of, the life cycle of the patent. Once you conceive an idea, you first execute it. One is ideation, which will be in mind, and then you execute it. You see the tangible product out of it. Then what will be the next step? Next step will be you have to do the patentability analysis on your own, wherein you would search different compounds which are available that are closer to your invented compound, whether exactly your compound is known or not. If it is not known, it passes the criteria of novelty. You will see whether your compound is having any advantages over the reported ones. If it is showing advantages, it will pass the criteria second also. So like that, each company, each institute has to do a preliminary patentability analysis on their own. Then they have to go ahead and file a patent application, do the patent prosecution in the respective patent offices. Once it is allowed, it is granted, they have to maintain it for the entire life cycle of the patent. Yeah, please move on to the next slide. So these are the specific steps in the ideation, the patent life cycle, snapshot, which I just explained. These are the further narrowing down of the steps. As I explained, in patentability analysis, you do the searches, you find out anything is known closer to your invention or not. Yeah, okay. Okay, so I will quickly take you to the case study. Can you please move to the case study 1? Yeah. So, as I told you, there is a process for getting a grant of patent, and there is a process for challenging the patent also. Now, case study 1 is, which talks about how you can fight with the patent offices to get a grant of it. Now, this is an example between Diamond versus Chakrabarti. So, Diamond is the commissioner of the patent office in US, and Chakrabarti was a employee in the General Electric. Now, what Chakrabarti did is, he invented a bacteria which could break down the hydrocarbons in the oil. No naturally occurring bacteria could do that. And definitely it is having an industrial application, because whatever oil spillage happens in the seas, it can easily degrade all those spillages. So in 1972, Chakrabarti went and filed patent applications in the US Patent Office, covering various aspects of the invention. One of the invention was covering bacteria as such. Process part, patent office allowed. Composition part, patent office allowed. But when they were trying to claim the bacteria per se, patent office said, no, it can't be allowed. We can't give a grant of the bacteria. Why? Because it is a living organism. Living organisms are not patentable according to then rules. Chakrabarti appealed that decision to the higher authority, and Supreme Court in 1980 given a decision, allowed the grant of the patent. The only takeaway was, your invention is living or non-living, it is immaterial. What is important is, it should be novel, it should have an inventive step. So since in this case, it was a living organism, it was made by human intervention. It is not something he has discovered that bacteria. No, he has done certain mutations in that bacteria. As long as human invention, human intervention is involved, there is an inventive part of it, patents can be granted. So this is an example where an inventor can fight with the patent office and get a grant of it. Second example is totally opposite of it, wherein a patent was granted and the public at large was getting disadvantageous position because of that granted patent. So this is way back in 1995, when in US, for one university, the patent was granted for turmeric, which we call as haldi. This patent was granted, so basically legal rights were given to the US patent. Then our CSIR, the Council for Scientific and Industrial Research in India, they have challenged the grant of this patent. They went ahead, they fought with the patent office, they fought in the courts. They have shown the Ayurvedic text and the scientific publications from the Indian heritage, wherein it was well known that turmeric had anti-inflammatory properties. Turmeric was already known since ages. After showing all this data, the US Patent Office revoked the grant of the patent. So it was a big success for India because we do not acknowledge, or sometimes we do not document it properly. But these are the things which are becoming boom in the Western world. And that's where the importance of protecting the traditional knowledge becomes very much important. So if I can continue with 2 slides, otherwise I'll wrap it up. Okay, so the key learnings, patents are definitely the backbone for the pharmaceutical sector. Give you legal rights, it drives innovation, it improves the medical sciences. So it is definitely an element. Distinguishes from the other products. So trademarks are also important. Copyrights protects the copyrighted material. Trade secrets is something, which you have to weigh. What is the benefit versus risk ratio in maintaining certain inventions as trade secrets? Not all inventions can be kept as trade secrets. Yeah, please move on. Patents, it acts as a catalyst for innovation. When you know that there is a possibility of legal enforcement, there is a possibility of monetary benefits, it drives you to invent something. It gives you right to enforce. You can prevent others from entering into the market for a longer period of time. And because of the patents, because of the incremental inventions, it helps in the incentivizing the development of the life-saving drugs. In nutshell, definitely patents are the most critical part of the pharmaceutical industry. And since you are the students of the research institute, I would say that whenever you see anything which is different from what is known in the art, please go ahead and secure a patent application for it. You never know when an industry can come to you and, try to take license out of it. Thank you. So if any questions are there, please, the forum is open for it.
Thank you, Dr. Shweta Kulkarni. Madam. Thank you, Dr. Shweta Kulkarni, for informative talk and exploring as the pharma patent for the improvement of global health care to implicate the development of life-saving drugs and showcasing as the case study, synthesis, and pharmaceutical formulations. Thank you so much. Now, I request Dr. V. Ravikumar, Professor and Head, Department of Pharmacology, MNR College of Pharmacy, to acquaint as the speaker of the second scientific session.
privilege to welcome today's resource person, Dr. Saket Ram Tirugula, currently serving as a research officer Ayurveda at the CCRAS National Institute of Indian Medical Heritage, Hyderabad. He holds BAMS, MD in Ayurveda, Dravya Guna, and a PhD also in Dravya Guna. And Dr. Ram is an CCRAS Young Scientist Awardee and has made significant research contributions, particularly in information communication technology for mainstreaming Ayush systems. He has been instrumental in developing the key digital resource such as Ayush Research Portal, the National Ayush Morbidity Codes and standardized terminologies, electronics portal, and various digitalized books on Ayurveda. Furthermore, Dr. Ram has actively contributed to the development of WHO international terminologies of Ayurveda and a traditional medicine module for Ayurveda, Siddha and Unani in international classification of diseases. His involvement extends to the health focus group by ITU and WHO, which has developed a benchmark guidance document for the usage of artificial intelligence in a traditional medicine, reflecting his commitment to the integrated modern technology advancement with the traditional medicine knowledge. So far, he has 65 research papers and 7 books to his credit. With a small introduction, I welcome the resource person, Dr. Saket Ram Tirugula, onto the dais. Please come, sir.
Check.
Hello. I barely see. Uh, Good morning. I think after Dr. Shweta's presentation, I have very little to offer, but anyway, I have been tasked to introduce what is the possibility of patents, especially in the herbal phytopharmaceutical side. So let me try. I'll quickly try to wrap up my presentation, and let's see if we can have a little interaction afterwards and as and when it is feasible. So I'm addressing young students of pharmacy here. I come from Ayurveda background. I'm not a core pharmacy person as per se. I'm a trained physician, but maybe serendipity, I had opportunity to work with pharmaceutical people for some time, and my specialization fundamentally deals with the sourcing of medicinal plants for various therapeutic usages. Next slide, please. So I need not repeat this. We are all aware how rich and how diverse this country is in terms of demography, in terms of raw materials, in terms of many, many natural resources. So that is a big blessing for us, and at the same time, we are also having a very big legacy of experience of using plants for medicine, for food in very, very diverse forms, which are from the natural sources of India itself and many other things which we have brought to India from various sources from hundreds of years. So we are very good at really like sort of understanding plants and also like sort of translating them into various uses, not only medicine, it's food, clothing, many, many other industrial uses. We are very good in that. We should be very proud of that. This is one of the rare countries on earth which is having this kind of an ability, and we are all a part of that. Next, please. So madam has already explained to you about what is a patent and all. I'm not getting to the details of it. So these are the cornerstones: invention, whether it's a product or a process; and you will get 20 years, and as madam has said, in exclusive cases, another 5 years of leverage, where you have the exclusive rights of like sort of monetizing it. That is the incentive part. So why patents are there? To encourage innovation, reward the inventors. This is very, very important. Invention and truth, both are very expensive. Nothing comes for free. So it's very utopic to understand, or say, if somebody says that you should give away things for free and all, good. On the face value of it, it looks very attractive, but at a practical level, that doesn't hold good. And as a matter of fact, the so-called developed countries are developed not just because of their military strength or things like that. They are developed because of the kind of wealth they have generated in terms of ideas and the kind of patents they have. For example, Israel, I'm taking few names. Israel is such a small country, but that is the leader of many, many patents. And that's the reason, irrespective of whatever the geopolitical situation is there, that country is indispensable. We cannot survive without a country like Israel producing good ideas, patentable ideas, and giving it for the humanity in a certain manner, also taking some benefit, but ensuring that we are having better technology, better facilities, better safety, and many things like that. That's possible only because of the hardworking inventors. Why US is so favorite for many people? Because they encourage innovation, they invest a lot in innovation, and also they market it very well. All the Fortune 500 companies, most of them are having some kind of origin in US. It can be any kind of a technology. It can be traditional technologies, it can be pharmaceuticals, it can be IT, you name anything. So wherever such innovation is possible, even the countries like, for example, Singapore, now China, they are there because of this encouragement in the innovation, and we are just running a little behind, but I think now we are catching up with that. You see, taking somebody's idea and mass producing it, doing service is also, that also will generate revenue. But if you have the original idea, you get much more revenue than what we envisage. So that's where I think today's generation should start invest most of the time. It's very, very important. We are just near saturation at the service level. All those things are occupied now. But whoever have the original idea has the future now. Next, please. So this is also, I think, madam has already explained. I'm not getting into it. Next slide. Next slide. So I'll directly delve into the traditional medicine part of it. This is very important because I'm starting with the bad news. If it is a traditional thing, it's a prior art, and you can't take a patent. Then you must be wondering why this person is invited to talk about. If you are not able to take any patent, what's the meaning of this whole session? You will understand it later. So this is a caveat. Means if you want to patent something, you ensure that this is not the obvious traditional knowledge. For example, how many of you know to cook food in at least in some extent? Please raise your hands. At least rice. Most of you, right? So you put some water, soak it for some time. So you understand it. You have learned it from either from parents or somebody. It's very obvious. Obvious not because of its like thing, because we have learned. The Neolithic people, the old nomadic people, cooking was an invention, right? But it has become so normal, now it has become very obvious. Today our job is to do something like cooking for an old caveman. If that invention is so novel, that is patentable. That's a fundamental thing you must all understand. So you just can't patent everything. You can just put a little sort of some external cover or something, just fill some new and the old bottle, or say vice versa, and say patent, that won't help. So that's why whenever you have a patentable idea, just double check whether it is already part of some traditional medical system. We have very rich traditional medical systems in India. Ayurveda, Siddha, Unani. They have such a big corpus. I will come to that. TKDN, madam has spoken about with respect to curcumin. I'll come there. Next one, please. So you have to, this is for filtering what can be patented. If you are attempting something with the plants or say plant-related medicines, so you must have this ability to distinguish whether it is a traditionally used one or if it's a new one. So that double check you always have to do. So Indian Patent Act 1970 and traditional medicine. So section 1004. So this origin, you have to disclose it. Opposition grounds it defines. I'm not getting into the technicalities of it, but part of an existing knowledge system. Next one. So wow, this becomes a big challenge. As I said, you have to filter a lot of literature and ensure that it is completely. It is very much doable. Luckily, we have a very good database now in the form of traditional knowledge digital library (TKDL). India is the only country in the world which has this kind of a library. So we should all be proud of it. It is a model example, though it is done for defensive patenting, but that also serves us to filter and come up with new ideas. For example, a simple example, in our most of the traditional medical systems, rest of the parts are not used. For example, stem is not used, leaf is not used. So that's a completely open area. You can try. And they also have very good pharmaceutical needs. It's not so complex as I'm speaking, but it's only a matter of keeping in mind that if it is directly traditional medicine, you can't take it. That's only the purpose of showing it. Next one, please. So inventive step is also there. If it's, for example, if it is a replica of existing process, in Ayurveda there are already some established practices, like for example, how to prepare a decoction in a certain way. We call it kashaya. So there is something called vati or say churna or say jutika. All those are traditionally available. So when you think of a patentable thing, it should be different from those things. That much of a care we have to take. Next one, please. Standardization is a big problem in traditional medicine because they are all natural substances prone for disintegration and they are all depending upon the plants and natural things which are sometimes sourced, sometimes collected, sometimes cultivated. So maintaining that batch consistency is a big challenge. It is not like a synthetic molecule. Synthetic molecules are per se developed in controlled environment, and if you have the process standardized, then it is done. You can have multiple batches of it, but in case of traditional medicine, it is difficult. So whenever you patent something, the industry will look at that aspect, whether if they purchase or if they take that patent from you, what kind of commercial viability they will have, whether they can sustain that production and continue that. All those things they will think. So whenever you patent, you also think of those things. Next one, please. So these are some of the regular, though I call it regular hurdle, it is better cross-check all these things. Indian Biodiversity Act is very strict. They are very, recently also we have seen that Gachibowli fiasco and all of that. So now it has become a very sensitive issue. We should be very careful whether it is falling under any biodiversity act. So first of all, better rule it out than repent later. After investing so much of money and time into repent, there is no point. Next one, please. So this is what Shweta has just mentioned. So this curcumin, turmeric, was a big problem. So this is all known in India, but in America somebody said it's a new thing, it's antimicrobial. They fought well, but not just fighting. What Dr. Mashetev and their team have done is that they wanted to solve this problem forever. So they created a wonderful library called Traditional Knowledge Digital Library, where we have digitized almost all formulations available in the Ayurveda text, Siddha text, Unani text, and Sovarikpa text. That's a huge wealth of knowledge which India has created. It's more than, I think, 2.5 lakh records by now. It is maintained by CSIR, TKDL library in Delhi. Next slide, please. So this is the library which we have. So if you consult them, they will tell you whether something is already available. So don't attempt that. That goes into the traditional. You can still manufacture it, but as Ayurveda drug or something else, but it is not patented. So you have to sell it as a proprietary or say like Shastric Ayurveda product, but not as a patented product. So for that screening, we have wonderful resource available with us. So for defense, and also this will help others patenting our obvious things in an undue manner, that also we can prevent. So many things are prevented. I think around 300 patents like that are prevented so far. Next one, please. So yeah, this is the impacted success. We are working with almost 5 major patent offices, and this is one of the rarest of the rare resources available in the world. I think no other country, only recently I think Malaysia has started, and I think Thailand also has started. But I think right now we have set an example to the world that we can protect our traditional knowledge through a very properly documented system. So next one, please. So now this is what actually when Dr. Vishaka called me and said, sir, you have to talk about payments, I was perplexed because traditional medicines are not patentable. But I gathered my courage to, of course, I knew that there is a new drug category called phytopharmaceuticals, recently introduced, very recent, I think 3 or 4 years old. That is a big gold mine for us. So I thought I think I can report this to students and generate interest so they can start thinking, whenever you think about a plant, you can now properly do all the toxicity, preclinical, clinical, if you do everything in a regular manner, what you all do to a chemical molecule, if you do it for a plant with having at least 4 active biomolecules and has a proper standardized process, that is now licensable under Phytopharmaceuticals Act. We have a very good category introduced, and I must tell you, India is the only country which has this provision right now. Other countries do have standardization processes, but for licensing and prescription, it is not possible. So, as I said, this is so though it seems little expensive, but it's the best way. If you go into the traditional medicine, you have some limitations. For example, if you produce an Ayurveda drug, only Ayurveda physicians can prescribe it. That's a small market comparatively. And to some extent, you can sell in the OTC. Export also becomes a little bit difficult. Or you have to, for example, like change it into a sort of food supplement or something. So they are all like sort of shortcuts. But now, having this phytopharmaceutical category as part of new drug entity, so we have the wide, bigger doors open for us. If you do little homework, complete all these steps diligently, we have a proper prescribable drug by any allopathy or any biomedical practitioner across India. Not only them, because it is a phytopharmaceutical, with some kind of a little bit of adjustment, even Ayurveda and other physicians also can prescribe this drug. So that kind of a new area has come now. So this brings
pharmaceuticals in several ways. So these are all different methods under which you can take patent. It's not just only the finished product or a direct marketable product. There are many intermediate steps. So every step is patentable. So for example, isolation and purification of novel compounds, you can patent it. New extraction or formulation process, you can patent it. This is applicable for even Ayurveda and Siddha also. So, for example, the same Chyawanprash, if you are able to cut cost and do it properly in a little modified process, which is, of course, in accordance with the textbooks and all, that is also patentable. And, of course, some examples are also there in such category. Next one, please. So, new therapeutic uses.
For example, during COVID, we have seen many traditional drugs we have repurposed for use of COVID. For example, in case of Ayurveda, there is CCRS has developed an Ayurveda drug called I-64 malaria. So we have repurposed it.
We have done trials and used it for COVID-19. So that's a
latest example we have. So that kind of a
repurposing also you do. So that is a novel indication for that. So that is a very good area for patents. Synergistic combinations. So for example, most of the cancer treatment, it goes in combination. For example, TB, it's a combination of 2-3 drugs. It's not single drug. So that combination novelty also is patentable. So that's a wonderful area. So modified herbal derivatives, this is another important area. Specific formulations and delivery systems. So it's just not oral or say topical. You can come up with some other novel drug delivery mechanisms also. So they are all patentable. Next slide, please. So in the international context, so we have many guiding agencies like WIPO and ICG and anyway, this is a theoretical part. We did not really delve into it. So they generally provide guidance and like coordinate between different patent offices. Next one, please. So these are the opportunities we have. R&D focused standardization methods, novel extraction, purification, synergistic combinations, new therapeutic indications, novel drug delivery systems, developing phytopharmaceuticals market. This is a very new area for India. We have to capitalize on it. Ensuring quality control and regulatory compliance, patent searching and analysis. So this is a specialist domain. I think Shweta madam and people, experts are there. We should take their help. It's not just a single person's job. It's a teamwork. We have to work with them. Otherwise, it's very difficult. It's a complicated documentation. Next one, please. So, for example, I have flashed this as an example. This is patent search on the Google. It's a very good database. Very good, very nice database, and almost 30-40 country patent offices information keeps on gathering, and you can search on patent.google.com. So that's a very good resource. You can just browse and just examine. It will give you the patent number and all those details. Next one, please. So to conclude, patenting herbal medicine in India involves navigating traditional knowledge protection, section 3P, patentability criteria, novelty, inventive step, and regulatory requirements. So CDCSO is the one who grants for this phytopharmaceuticals. You should not come to Ayurveda licensing people. That's a bad step. If you are having a phytopharmaceutical with 4 bioactive molecules, you have to directly go to CDCSO like any other new drug molecule development. Remember, don't waste your time approaching the like Ayurveda licensing agencies. There is no point. So TKDA is a vital defensive tool against bioprophecy. Of course, not only a defensive thing, it also helps you understand the novel aspect of our own health. So in that way, we can cut short a lot of time. So now, of course, till recently there were some restrictions of use of TKD, but now they are allowing it by certain fees or something, some subscription model they are coming up with. Once that is there, we can search this database. So through innovation, new process, use synergy, as I'm repeating again, formulation beyond existing traditional knowledge is key for obtaining patents. Phytopharmaceuticals offer a regulated pathway for science-based botanical drug development. I'm emphasizing, please focus on this part. This is very, very, very new venue the government of India has opened up for all of us. Finally, huge potential lies in scientifically validating and innovating upon India's rich traditional knowledge base. Finally, next slide. Thank you so much for patient hearing. Thank you very much. Namaste.
Thank you so much, Dr. Saket Ramachindra, for explaining us the major challenges for patenting and novelty in patenting, which gives a glance idea of our patent. Thank you so much. Will be having a formal inauguration program. So meanwhile, if you have any queries, you can raise your hands. Mic will be pointed to you. Program of one-day national conference. The one-day national conference on advances in drug discovery, development and patenting, challenges and opportunities aims to bring together researchers, practitioners and policymakers in the field of pharmaceutical science to explore the latest advancements and challenges in drug discovery, development and patenting. The conference will provide a unique platform for sharing knowledge, professional growth, fostering collaboration, and addressing issues with the pharmaceutical industry.
The pharmaceutical industry plays a crucial role in improving global healthcare. It requires integration of scientific research, clinical testing, regulatory approvals, and intellectual property management. This conference will offer valuable insights into the multidisciplinary approach needed for successful drug development and patenting, focusing on both opportunities and challenges.
MNR Educational Trust begins its journey with a commitment of quality education in 1974 by our chairman Shri M.N. Raju sir. The trust has come a long way, playing a pivotal role in shaping the educational landscape not only in Telangana but also in other states and overseas as well.
Sri M.N. Raju sir's
Noble aim to enrich lives through education and shape the students into proud citizens of the country, and it's been realized through the 78 plus institutions and hospitals. As the baton is passed, the Vice Chairman of the Trust, Sri Ravi Varma Mantena sir, is carrying on the legacy with great passion by accelerating the growth of the Trust with his innovative and exemplary leadership skills. His wise, objective outlook has led to the successful establishment of the MNR University.
The MNR Educational Trust remarkable and impressive journey for the past 50 years has been celebrated on 20th December 2023 at Parade Ground, Secunderabad. The President of India, Srimati Draupadi Murmu, graced the Golden Jubilee celebrations of the Trust.
There was a special ceremony honoring our chairman, sir, by the president, by Chairman Sir M.N. Raju, sir, and by the President of India, and he has been felicitated by all the pillars of the institution who has seen MNR through its formative years and nurtured it to its current stature.
With the visionary approaches of its Chancellor, Sri Ravi Varma Mantena, MNR University has become a 21st century world-class overall higher education and skill capability provider to meet the global challenges.
The MNR University is a multi-institutional and multidisciplinary university, making curriculum more inclusive and offering skillful education for increasing employability potential of the graduates and postgraduates. The university envisions enabling students to have access to world-class futuristic university and to provide a holistic environment to impart knowledge and skill sets to face global challenges in the future.
The MNR University aims to educate, encourage, and enable individuals to develop character, ethical values, knowledge, intellectual curiosity, and scientific temper across a range of disciplines of technical education, agriculture education, healthcare education at one place.
MNR College of Pharmacy, located on the MNR Hera campus at Sangareddy, was established under the MNR Educational Trust in the year 2004. And since then, it has been nourishing the pharmaceutical profession. It is one among the 75 renowned institutions of MNR Educational Trust. The MNR College of Pharmacy is approved by the Pharmacy Council of India, and it has been accredited by NAC and is affiliated to Osmania University, Hyderabad for all its courses, which are B.Pharm, M.Pharm in Pharmaceutical Chemistry, Pharmacology and Pharmaceutics, PharmD and PharmD Post-Baccalaureate.
Along with academic excellence, MNR College of Pharmacy excels in extracurricular activities which includes sports and cultural competitions, entrepreneur and communication skill development programs and competitions, career and competitive examination guidance, as well seminars in research and development. The institution encourages social activities through its National Service Scheme and Institution Innovation Council for the holistic development of students.
The college possesses contemporary laboratories providing best platform for learning students under the guidance of Dr. V. Alagarsamy, an esteemed personality in the field of pharmacy and research.
Dr. V. Alagarsamy is one of the top 2% scientists of the world for the 5th consecutive years by Stanford University, USA. Dr. V. Alagarsamy has received Young Scientist Award from the Department of Science and Technology and was honored with a Fellow of Royal Society of Chemistry, London. He has authored 10 books, 2 patents, and more than 200 publications and many other achievements to add on.
Dr. V. Alagarsamy, with his continuous guidance, has motivated all of us to reach this momentous day.
It's our privilege to invite our esteemed guests onto the dais to grace the special occasion with their presence. I could like to invite Professor Dr. V. Alagarsamy, Principal, MNR College of Pharmacy, Director, MNR Foundation for Research and Innovations, onto the dais.
Now we would like to invite Sri Ravi Varma Mantena, Vice Chairman, MNR Educational Trust and Chancellor of MNR University, onto the dais.
Being the Vice Chairman of the Trust brought a tremendous responsibility on his young shoulders. He has embedded the legacy of Sri M.N. Raju and is working for the holistic development of MNR Empire. He is an enthusiastic and a cognizant leader known for his innovative and exemplary skills. We are honored and privileged to invite the distinguished individuals onto the dais.
Inviting guest of honor, Dr. Saketram Tirungula, Research Officer, Ayurveda, Central Council for Research in Ayurvedic Sciences, National Institute of Indian Medical Heritage, Hyderabad, and former OSD, Ayush Grid Project, Ministry of Ayush, Government of India, to grace the dais with his presence.
Next, we invite Dr. Shweta Mahajan Kulkarni, Associate Director, Intellectual Property Management, Dr. Reddy's Laboratories Limited, Hyderabad, to grace the dais with her presence.
Inviting Chief Guest Dr. C. Mallareddy, Professor, Department of Chemistry at Indian Institute of Technology, Hyderabad, to grace the dais with his presence.
Now we request Sri M.N. Raju sir, Chairman MNR Educational Trust and President MNR University, to come on to the dais. Sri M.N. Raju Garu, philosophy is to enrich lives through quality education and shape them into complement and dignified citizens of this great nation.
On this occasion, we would like to thank and welcome Dr. Manilal sir, Director of MNR Homeopathic Medical College and Hospital, for coming. Also thanking Dr. Revijilakala sir, Academic Director. Thank you for your presence, sir. Thank you, Manikandan sir, also for coming to this occasion. The function of prayer is not to influence God, but rather to change the nature of one who prays. Prayer is a process of purifying our souls. Let us evoke the blessings, guidance, and wisdom of the Almighty through the prayer song. We invite Mr. Om Prakash of PharmD second year to recite the prayer song.
నిషగ నిషగ శని సగరి సని ద మగరి శని దబ మగరి శని ద మగరి శని దబ మగరి శని ద మగరి శని దబ మగరి మహాగణపతిం శ్రీ మహాగణపతిం మనసా స్మరామి మహాగణపతిం మనసా స్మరామి మహాగణపతిం మనసా స్మరామి వశిష్ఠవామదేవాదివందిత మహాగణపతిం మహాదేవసుతం గురుగుహనుతం మహాదేవసుతం గురుగుహనుతం మహాదేవసుతం గురుగుహనుతం మారకోటి ప్రకాశం శాంతం మహాగణపతిం మనసా స్మరామి వశిష్ఠవామదేవాదివందిత మహాగణపతిం एकदंत दयावंत चार भुजाधारी एकदंत दयावंत चार भुजाधारी माते सिंदूर सोहे मूषिकी सवारी माते सिंदूर सोहे मूषिकी सवारी एकदंत दयावंत चार भुजाधारी माते सिंदूर सोहे मूषिकी सवारी पान चढ़े फूल चढ़े और चढ़े मेवा पान चढ़े फूल चढ़े और चढ़े मेवा लदन को भोग लगे संत करे सेवा जय गणेश जय गणेश जय गणेश देवा माता जाति पार्वती पिता महादेवा माता जाति पार्वती पिता महादेवा माता जाति पार्वती पिता महादेवा थैंक यू
Thank you, Om Prakash. Brightness and prosperity are the symbols of light. It is a positive force that removes darkness. If once we feel that how terrible the darkness is, then we could crave for the light with our real essence, because it shows the right direction towards our bright future. I take the privilege of requesting our esteemed dignitaries to light the lamp and mark the auspicious beginning of the day. We request all the dignitaries to join for the lamp lightening.
बुद्धि
complains about the wind, an optimist tries to change it.
and a leader adjusts the sails. The sails of this particular notion were adjusted by a person who is not only a great motivation to all of us, but a visionary and the person who gave us the belief to bring together this event. We request Principal of MNR College of Pharmacy, Dr. V. Alagasamy sir, to welcome the concourse.
Good afternoon, dignitaries on the dais, off the dais, and my dear students. Work is worship. Work is worship. This is the slogan given by our chairman, sir, and all the staff members, faculty, students, all we follow this slogan in this campus. That's why today in this morning, without bothering the formal inauguration, we have started the scientific session. We follow the slogan, energy, ideas, whatever given by our chairman, Sri M.N. Rajgaru. Sir, we are always marching ahead and surely we'll make the MNR institutions great in this country coming days. Your valuable insights that you are extended to all our students and faculty members always energize us, and your presence surely it will be a great enthusiasm for us to make these young students and the delegates in their career a successful one. So we all welcome you onto this national conference wholeheartedly and energize and bless us to have the successful careers. We have the teammate or the co-member of the working force of the MNR educational institutions. That is nothing but our Vice Chairman and the MNR University Chancellor, Sri Ravi Verma Garu. He is always work among us faculty members and the non-teaching staff members, and he is always a backbone for all the working peoples in the campus. It's our pleasure to have you in this August gathering, sir. We welcome wholeheartedly to this national conference, sir. Today's chief guest, Dr. C. Mallareddy Garu, the professor of IIT Hyderabad, who is well connected to the pharmacy department, that is the organic chemistry and the drug discovery. So the drug discovery, the conference title is coincide with the interest of the research that is doing by some of the IIT professors in Hyderabad. So when we searched the Google, we found one of the eminent in the department. So we approached Dr. C. Mallareddy Garu. He immediately accepted our invitation, and without any hesitation, he is ready to address our students in this national conference. So on behalf of everyone, we wholeheartedly welcome you to this conference, sir. We have presently all Ayurveda, homeopathy, and Indian system of medicine is developing in the whole world because of the many of the adverse drug reactions that we are coming up with the allopathy medicines. In such a way, the herbal medicines becoming very much important. So, Dr. Saket Ram, who is with us, who is one of the eminent in the Ayurvedic medicine, he has been accepted to deliver a lecture in this August gathering. So, we welcome you to this conference, sir, on behalf of everyone. Whatever the inventions we do, we have to patent. If you don't patent, it will not be of your property. So, Dr. Sweta is with us to patent all the inventions that we are doing in the pharmaceutical industry. She had given a wonderful lectures in the first morning. So, we all welcome again to this conference, madam. In addition to all the dignitaries, there are so many eminents and the faculty members from the different colleges, and we also have the experts of the various authorities of the MNR Higher Education and Research Academic Campus. We wholeheartedly welcome one and all. We see so many delegates from the different pharmaceutical industry and so institutions. from the different colleges of Hyderabad especially. We hope the MNR College of Pharmacy always stand in one of the top grade institutions, and the academics what we do always be in the top class. We never compromise with the quality of education, because our management always sees the discipline and education as two eyes. We maintain the discipline and the education and the quality of academic achievements and the research what we have showcased in the pharma fraternity always inspire all the young and budding students who are visited to this campus. I hope we will not disappoint you for coming over here, and surely we will make your professional interest to grow higher and higher. And all our faculty members who are working day and night to make all your interest to do well in this conference. I hope all you will enjoy this stay at one day, and will not waste our time, and will not feel you to disappoint in this profession. And we have all the experts, and even we have the many of the faculty members who are expert in their departments. You can interact with us, and in coming days we will be having more than 2 days conference also. That day you may leisurely you can interact. So thanks for coming all the way, and we welcome one and all to this conference and make you this conference a great success. Thank you.
Thank you, sir. Now, nothing great will be achieved without a great mentor, and mentors are only great if they are determined to do so. To carry on the legacy and fulfill the vision of the trust, Sri Ravi Verma Manthana sir, Vice Chairman MNR Educational Trust and Chancellor MNR University, adds a perfect blend to modern approaches under his dynamic leadership. I request Sri Ravi Verma Manthana sir to address the gathering.
So a very good afternoon to all. In particular, our chief guest, Mallareddy Garu, Saket Ramji, and Sweta ji, our chairman sir, Principal Dr. Algar Swami, all the dignitaries of the dais, and all the faculty and students from various pharmacy colleges in Telangana, and also from MNR Pharmacy College. Welcome you all for this wonderful event. And I really appreciate the efforts of Dr. Alagar Swami and his team for organizing this kind of wonderful session, because this makes our students think beyond classroom, not just their curriculum and other things, but this will allow them to think beyond classroom. And the eminent speakers who are there, one is from chemistry background, one is from Ayurveda background, and one is from intellectual property rights. So they are all stalwarts in their field. So they have almost, I think 2 sessions are already completed. One more session is pending, I think. One more session is due. So we are not actually competent to speak in this forum, because we are not from any of these backgrounds. We are pure administrators. But since I have been called, one advice I want to give to most of the students is that you need to focus on research. That is the most important thing. And out of thousands of pharmacy graduates coming out every year from all over the pharmacy colleges of India, very few are getting into research. Maybe less than 1,000. So this, unless we get into research means not just doing PhD or just publishing some publications in the journals, not that one, but what the advanced countries are doing. Try to understand what they are doing. They are trying to not copy from anything. They are trying to invent something new. And for that kind of inventions, they spend years together, maybe 5 years, 10 years, or entire life they dedicate for bringing out one product or one patent. So this takes a long time, there is no doubt about it, particularly when you are dealing with human lives and research on medication related to human beings. It takes a long time, there is no doubt about it. But that one invention can change your total life. And people who are thinking of earning money and other things, that is not important. First of all, if you are knowledgeable, if you are doing something great, if you are able to bring out a product, or if you are able to contribute towards research, automatically money will follow you. There is no doubt about it. So try to concentrate on doing research, and we are there to support you in all the ways, whatever way we want. We are ready to bring in some very eminent people into the campus to help you, to guide you towards research. We are always there. And apart from that, we have recently acquired the status of university. That is, now we are MNR University. And last August we got the approval. And under the university also, we are concentrating more on multidisciplinary courses, interdisciplinary courses. And like pharmacy students can also have knowledge about artificial intelligence, because now AI has become a buzzword. Everywhere people talk about AI, and every field AI is going to influence. There's no doubt about it. So people need to understand about what is AI and how does it function, how does it relate to your particular field. So for this, we have already enrolled some students, particularly our medical students, our nursing students, our pharmacy students. They have enrolled for this short-term AI course, and they are already pursuing it. I was really surprised that there was huge demand for that course. When we initially wanted to introduce that course to medicine students and pharma students, we were a little skeptical about whether there will be response or not. But there is huge response. So that means students are also thinking in the direction, not only about their particular course or their particular curriculum, they are thinking beyond that. That made me feel very happy. So in this direction, we want to go for many such things. Even for intellectual property rights, you need to know about what is IPR. So we will have a short-term course on IPR also for you. Already we have been conducting the IPR courses also here. And we will have some short-term course on IPR also. Like that, we can bring out very innovative things. And right now we have 3 institutions under the university. And in future, we are going to start a management school. Right now we have School of Technology, School of Medical Health Sciences, and School of Physiotherapy Rehabilitation. And in future, we want to bring in School of Management, School of Law, and wherein all these institutions will be offering interdisciplinary courses, multidisciplinary courses. And thanks to National Education Policy 2020, now a student can pursue 2 undergraduate degrees simultaneously. That is, if you are a pharmacy, you want also to have a degree in management, you can do 2 degrees simultaneously in the same 4 years period. So that's allowed now. Similarly, an undergraduate student can directly apply for PhD, no need of doing masters. Similarly, interdisciplinary courses or this integrated programs, that is B Pharmacy with MBA, 5 years course, that's possible. So all these things we want to bring in to this campus, and we want to facilitate our students in this very fast growing the society. We want to bring in our students to be very competent with this kind of integrated courses which can help the students to progress in their career. So once again, I don't want to take much time. I thank the guests who have come here to deliver the lectures. I thank them for sparing their valuable time and coming over here. And we look forward for more such events to happen in MNR College of Pharmacy. Thank you one and all.
Thank you, sir.
The fragrance of flowers spread only in the direction of wind, but the goodness of a person spreads in all directions. With the continuous inspiration and guidance of our founder, Sri M.N. Raju sir, MNR Educational Trust envisions instilling leadership skills and overall personality development among students and faculty alike. With great pleasure, inviting Sri M.N. Raju sir, Chairman of MNR Educational Trust and President of MNR University, to say a few words.
I wish great grand success for the day of one-day national conference. I wish all the participants
On behalf of our chairman, sir, I can say, chairman, sir, used to talk at least 10 to 15 minutes. That may be the best speech always. But even he is physical unable to come, but he wants to see the gathering and he wants to see the students like Abdul Kalam, how he always get energy from the students. He always join with the students. So we, on behalf of the whole gathering, we thank you, sir, for coming over here and blessing us and supporting us. Thank you, sir. Thanks a lot.
Thank you, sir. Now let's move on to the memorable part of our program, the release of our souvenir. This beautifully designed proceedings is a tangible reminder of our conference, capturing the essence of our gathering and serving as a memento for all the participants. We request all the dignitaries to come forward for the release of proceedings. We request We request all the dignitaries to showcase the proceedings of the one-day national conference on advances in drug discovery, development, and patenting challenges and opportunities 2K25.
Now is the time has arrived to add some fragments onto our dais. A country's rich culture will always be present in all parts of our lives. Therefore, even in this new era of technology, a shawl is adorned to the dignitaries and it is considered a rich felicitation present and signifies respect and gratitude. We request all the dignitaries requesting all the dignitaries to felicitate the chief guest of today's one-day conference, Dr. Malla Reddy sir.
Our chief guest, Dr. Sri Malla Reddy, Professor, Department of Chemistry, Indian Institute of Technology, Hyderabad.
Requesting the dignitaries to felicitate today's guest of honor, Dr. Saikat Ram Tirugula.
Dr. Saket Ram Trigula, Research Officer, Ayurveda, CCRAS, MH, Hyderabad, OSD, former Ayush Grade Project, Ministry of Ayush, Government of India.
Requesting the dignitaries to felicitate Dr. Shweta Mahajan Kulkarni.
Our next guest of honor, Mrs. Shweta Mahajan Kulkarni, Intellectual Property Management, Associate Director, Dr. Reddy's Laboratories Limited, Hyderabad.
Requesting the dignitaries to felicitate our beloved chairman, Sri M.N. Raju sir.
Our chief patron, Sri M.N. Raju sir, Chairman, MNR Educational Trust and President of MNR University.
Requesting the dignitaries to felicitate our chairman, Sri Ravi Varma Mantena, sir.
Patron Sri M.S. Ravi Varma, Vice Chairman MNR Educational Trust, Chancellor MNR University.
Felicitating our Vice Chairman, sir, Sri Ravi Varma Mantena, sir. Vice Chairman of MNR Educational Trust and Chancellor of MNR University. Next, requesting the dignitaries to please felicitate our principal, sir. Dr. V. Alagaswamy, sir, Professor and Principal, MNR College of Pharmacy, and Director of MNR Foundation for Research and Innovations. Thank you all dignitaries on the dais. Requesting all the dignitaries to occupy the chair among the audience. Now we go for the third scientific session of the day.
Now I request Dr. M.D. Sultana, Professor, MNR College of Pharmacy, to acquaint the speaker of third scientific session.
Thank you. Live as if you were to die tomorrow. Learn as if you were to live forever. Mahatma Gandhi. Good afternoon to all dignitaries on the dais and off the dais. Welcome to today's conference. Myself, Professor Dr. Sultana, and it's my pleasure to introduce our today's chief guest, Dr. C. Mallareddy, Professor, Department of Chemistry, Indian Institute of Technology, Hyderabad, who has graciously accepted our invitation to join us today. Professor Mallareddy is a prominent scientist in the area of supramolecular chemistry, crystal engineering, solid-state pharmaceutical chemistry. He earned his postdoctoral fellowship in the Institute of Nanotechnology, Germany from 2007 to 2008. His academic career includes leadership and diverse teaching positions at various institutions, including Indian Institute of Science, Education and Research, Kolkata, and various visiting positions at Karlsruhe Institute of Technology, Germany. Honorary Professor, University of Bradford and University of Leeds, UK. Dr. Mallareddy has been received various awards such as Fellow of the Royal Society of Chemistry, Bhagyadara Award from Punjab University, Swarna Jayanti Fellowship in Chemical Sciences from DST, Government of India, and received Distinguished Lectureship Award from Chemical Society of Japan, that highlighting his international status in research and development. His multifaceted contributions encompass research, education, leadership, and advocacy in research, making him a respected figure in academia. He received 14 funded projects from various prestigious funding agencies. Professors Dr. Mallareddy's research contributions are significant as demonstrated by his publications in highly indexed and peer-reviewed journals. His work has received national attention and has made a tangible impact on communities both locally and globally. He is guiding more than 15 PhD students. We honored to have you, sir, today with us. With this brief introduction, I would like to invite our distinguished chief guest, Professor Dr. C. Mallareddy, to address our students and researchers. Sir, we are eager to benefit from your wisdom and insight. Please take the stage and share your valuable thoughts in chemistry and research and drug discovery with us. Thank you, sir. Once again, I welcome you.
Can you? I can't. Can you move it to this side? Because normally without seeing the audience, it will be difficult for me, but it's okay. One is, I think, okay. Yeah. Okay. Yeah. Good afternoon, everyone. Yeah. So it's a great pleasure to be here, and probably I have never addressed so many audience. So probably this is a record for me personally. So it's really wonderful to see all of you here. So I am given around 20 minutes. Okay, so I am also hungry. So I will not take more than 20 minutes. I will try to, but at the same time, I wanted to make you understand about crystals. Okay, these are everywhere. So before I finish, I will make sure that you understand what crystals are, and then you will start seeing them around you from today. Okay, so that's what I try to do today. So we all know that of course, when you study material science, we obviously see crystals everywhere. Okay, but probably if you do not know the chemistry, for example, chemistry is the subject which brings good chemistry between various disciplines. So it is very important to understand chemist's perspective. So today I wanted to talk about crystals and then connections with the medicines. Okay, that is what I actually try to do it today. Can you go to the next slide? So before I go into the topic quickly, I want to thank all my PhD students, present students and former students, postdocs who have done research work in my lab, and then that is what allowed me to basically contribute to different fields. So then I also thank all the collaborators. If you want to really publish at the international standard, okay, if you want to compete at the international level, so you have to make sure that whatever you discover has a larger implication. Okay, you have to put it in a larger context. For that, collaborations are very important, because if you do something in your own area, okay, you can be expert only on a small subject, but you cannot be an expert on a very large subject. So it is very important to collaborate with other groups, then your work will actually become much more wider, and then implications also can be understood by more people. That's why it is very important to collaborate. And also I am editor-in-chief for this Christian Come journal. So we publish, authors publish papers on the pharmaceutical solids and many areas. So can you go to the next slide? So this is a chemistry building at IIT Hyderabad, and if you closely observe, it looks more like a periodic table. Okay, so each building has its own design. So this is a chemistry building, and can you go to the next slide? And so here you see my group in IISER Kolkata. I worked for 15 years at IISER Kolkata. Then I recently moved to IIT Hyderabad, and that side you see nice guest house and convention center where we do the conferences at IIT Hyderabad. So that is IIT Hyderabad building. Go to the next slide. So now I have a question to you. Can someone define a crystal? What is crystal? Can someone define crystals? What is a crystal? Any idea? Okay, go to the next slide. Next slide. Next slide. Yeah. Can you recognize this? What is the structure here? This is the structure of. So each sphere is a carbon atom. Each carbon atom is connected to 4 other carbon atoms with a tetrahedral geometry. What is this structure? Anyone? There is a precious jewel. What is that? Diamond, right? So this is a structure of diamond, right? So diamond is a best example for crystal. Is it clear? So in a crystal, atoms are arranged in a regular fashion. So that is what is called crystal. Okay, go to the next slide. So now can you see here? What do you see here? Regular arrangement of people, right? So you can say this is a reminiscent to, or you can compare this with a crystalline arrangement. Okay, so when you see a repeated arrangement, then of the same units, so you can compare it with a crystalline structure. So in a crystal, molecules or atoms or ions are arranged in a regular fashion the way you see people here. Okay, so this is how the molecules are arranged. Go to the next slide. So what do you see here? It's not a crystal, right? So if it is not a crystal, you may have still the same contents, but you don't have regular arrangement, so then you can't call that as a crystal. So if you have a material and if the constituents are not regularly arranged, so you can't call it a crystal. Then what do you call it? Anyone? What kind of material is it if it is not regular in terms of arrangement? Yes, ma'am, you are not allowed. you will patent it, right? Yes, please. Amorphous material. If it is not a regular arrangement, you call this a amorphous material. Okay? So which one will be more stable? Crystalline material or amorphous material? Perfect. Very good. You are all good chemists. Okay? That's great. So there is lot of meaning to this. Okay? So if something is very stable, okay, then molecules or atoms are very nicely optimized and they go into a very stable structure. Once it is stable, then it will remain in that condition and it will not come out from that condition. So this has lot of implication to medicines. When we eat it, right, any tablet, so it has to dissolve and then it has to go at a certain dissolution rate, and then that has lot of implications to how this will actually act on us, right. So there is a direct connection to crystallinity and amorphous nature of the material. So I will try to introduce this concept using very simple examples, and if you understand it, I think my job is done. Okay, that's what I will try to explain in a very, very simple language. So why crystalline material versus amorphous material is important for medicines? When you make a medicine, synthetic chemists, our head of the principal is very good at making molecules. Okay, but after making molecule they forget about it, but then solid state chemists, those who understand the crystallography will come into effect. So then we can actually modify the structure in such a way that the properties, kinetics, dissolution rate, all these will be and can be modified. So that is where what I am going to talk about today is important. Can you go to the next slide? So you see, this is again a very simple slide. So you see 3 different types of states. What is that state? Solid and also crystalline. So this is liquid, right? This is gaseous state. Okay, this is the structure of the states, right? If you look at atomic level, so this is how we should be able to imagine. Good. Can you go to the next slide? So when you see them, no, the previous one. When you see them in real, like atomic scale, so crystal is never a frozen structure. Atoms always have thermal vibrations, right? As you can see there, it's a crystalline material, okay? But still atoms have certain vibrations, but they stay in their own position. They don't move and they are arranged in a repeated fashion, okay? Because it is a crystal. So in liquids, there is a lot of degrees of freedom, they move everywhere, but they are constrained under certain container or somewhere, wherever you keep them. Then gases have a lot of degrees of freedom, right? So this is a thermodynamically stable state, right? So all the materials will always try to go into the thermodynamically more stable state. Go to the next slide. So I'm sure again you can figure out what I am showing here. What is that? That's a diamond structure. So what is this? Graphite, right? So element is same in both of them. Both are made up of carbon element, but the structure is different. If you know, diamond is one of the stiffest materials. If you have to cut something, you use diamond. Why? Because it is a very stiff material. You can cut any material with diamond, okay? Graphite is very, very soft. We use it in pencil nib, right? If you rub it, the layers will peel off because there is no connection between these two layers. There is no covalent connection. This is a connection made by non-covalent interaction, pi stacking interaction. Just it is a kind of a van der Waals type of interaction. That is why the structure, because of their structure, so that is because all the carbons are connected with 4 other carbons, right? It's a covalent bond. So atoms are very stiff, that is why. But here there is a weak connection between the layers, right? So that is why it is very soft. You can move these layers very easily. So this tells us that internal structure of any solid will determine its properties, right? Whether a material will be soft or a material will dissolve very fast, where exactly it will melt, okay? All these properties will depend on the internal structure. So as you can see from here, so what do we call these two states of carbon? Allotropes, wonderful, right? These are allotropes. So structure is different, although element is exactly same, right? Go to the next slide. So as you can see, properties are very different. Go to the next slide, maybe you can roll. So same element, but the order is different, properties are also different. Go to the next slide. So there are many examples of crystals, right? This is a salt, and then we like crystals, but you don't want crystals in your kidney, right? So you are all going to treat patients. So we all like the crystals, but you don't want them in your kidney. So what you see, these stones are also crystalline, material, right? So these oxalates normally basically crystallize when you have a lot of concentration, right? So at higher concentration they crystallize, and then at lower concentration there will be a dissolution, right? That is why when you have stones, doctor will ask you to drink a lot of water, okay? Because in a lower concentration, the crystals will actually start basically dissolving. That is the principle, right? Go to the next slide. So go to the next slide. There are many types of crystals. All metal, metallic materials are also crystalline materials, okay? Then there are materials like, diamond. This is a covalent crystal. All the bonds are arranged with the covalent connections. And there are also molecules, okay? Molecules have all the covalent connections within the molecule, but between the molecules you don't have covalent connections. So you have non-covalent connections. That means simple hydrogen bonding interaction or electrostatic interaction between ions or very weak van der Waals type of interactions. So these are very, very weak interactions. Most of the pharmaceutical solids, okay, we call them as active pharmaceutical ingredients, short form API, right? All the APIs are small molecules, okay? So when they crystallize, they have to crystallize through which bonds? Non-covalent bonds. So weak interactions, right? Hydrogen bonding interaction or electrostatic interaction if it is ionic, or simple van der Waals interaction. These are the three different types of interactions through which these molecules crystallize, okay? So go to the next slide and go to the next, yeah. So these are different types of crystals. Even proteins can also form crystals, even rocks, okay, sand, everything is crystalline. Around us, most of the material except the biological materials, even in biological materials, our teeth, for example, bone is highly crystalline, okay? But muscles are not crystalline, but there is a great order at a molecular level. You still have order, but they are not perfectly crystalline, okay? So crystals are everywhere, even salt, table salt, sugar, all these are examples of crystals which are everywhere around us, right? Most of the materials around us, solid-like materials are actually crystalline, okay? So go to the next slide. Yeah, so these are some examples, zeolites, right? Aluminum silicates, right? All these are actually examples of salts. All the rocks, whatever sand we see around, 99% of them are actually completely crystalline. They are not single crystalline, but they are polycrystalline. So domains, basically grains will have different orientations. So but single crystalline means all the arrangement everywhere is exactly arranged in a same orientation. If you change the orientation, although it is crystalline, so you call that as a polycrystalline, but still it is a crystalline material. Go to the next slide. So all the pharmaceutical solids are mostly crystalline. Okay, you see them like white powder. White powder, whenever I ask students, what is this white powder? So they say that this is amorphous material. Amorphous means there is no internal order, everything is arranged randomly. If you see something like, white powder, doesn't mean that it is amorphous. If you put it under scanning electron microscopy and magnify it and see it, you will see beautiful small crystals, maybe micrometer scale or nanometer scale. 99% of the materials, organic materials are crystalline. Even pharmaceutical materials which you have inside the tablet, which looks like white powder, most of the time it is crystalline. Why? Because crystalline state is the most stable state. If you want to have amorphous material, you have to work hard, and that is very difficult, and keep it in amorphous state is very, very difficult, and that is where patenting will come. Amorphous materials are less stable, so they will dissolve much faster because bonds are not very strong, right? They are randomly oriented, so they dissolve much faster. Suppose if a drug has very, very poor solubility, okay, it may have great potential, great activity at a molecular level, but only it will act when it dissolves and goes into the body. But if it doesn't dissolve at all, even if you take 500 mg, only few mg go into the blood, for example, so it won't actually work. So you have to make them as amorphous material, okay? So there are many strategies to improve this. So you have to work at molecular level and change this crystalline state, then you will be able to deliver the medicines better, right? So that is one subject which is called as pharmaceutical solid state chemistry. Okay, this comes in the formulation of APIs. Once you make the molecule, once you have the solid, you can actually change that to improve physical properties, stability, thermal stability, photostability, hygroscopicity, or dissolution, kinetics. So many things can be modified using solid state chemistry. Go to the next slide. So yeah, just go ahead. I think you can skip this. We'll try to save some time. Yeah, go to the next. So go to the next slide. Yeah, so this is, as you can see here, there are two types of crystals here. So both the crystals belong to the same molecule, okay, same pharmaceutical solid, but they look different, okay. So go to the next slide. Yeah, so when you take API, for example, a molecule or pharmaceutical drug, so inside the packing could be completely different, although the molecule is exactly same, formula is exactly same, okay, but the way they form the interactions are slightly different. For example, you take bricks, you can arrange the bricks in multiple ways. But brick is same, single unit is exactly same, right? But you can organize them in multiple ways. So you remember the carbon allotropes. Structure is different, but element is same, right? So here also, in pharmaceutical solids, molecule is same, API is same, but the way they organize in the solid state could be different. When you have that, what do you call this? You call them as polymorphs. Okay, those are polymorphs. So the physical properties will be exactly same or different for polymorphs. Structure is different. In one structure, maybe interactions are stronger than the other structure. So all the physical properties will change. If you have more thermodynamically stable one, it is good, but it may have lower solubility. Okay, because molecules may not want to come out. So if you have to improve the solubility, then you may have to take the less stable one. If you get a metastable one, so that will be much better. If you can actually stabilize the metastable one, and then that will be much better. So pharmaceutical solids can have multiple states. Go to the next slide. So yeah, so you have a thermodynamically stable one on that. So this is the energy diagram for it. So you have a solution. In solution, you have your pharmaceutical solid. When you crystallize it, maybe energy barrier is higher for the thermodynamic one, but for the less stable one, if the energy barrier to go into the less stable one is lower, so maybe you will actually get a kinetic one. So different solvents may actually change the activation energy. So depending on the solvent conditions, you will be able to get either thermodynamically stable one, or you will be able to get either kinetically stable ones. Once you put the molecule into the solvent, so this scenario can change, but energy of the particular solid form will not change. Once it crystallizes, it will remain same. So crystallization can be one method to get exclusively one particular form. So here, those who work on formulation, those who come with the chemical engineering background, okay, they work on the crystallization. They try to control the outcome of the polymorphic form. If you want thermodynamically less stable one, which normally will have higher solubility, much better. So if you want to prepare that, you can actually use this energy diagram, change the activation energy by changing the conditions, and exclusively you will be able to control the outcome. So you will be able to get a better solid state form of the same drug. Drug is same, but the internal arrangement is different. So if it is metastable, it will have better properties. So all this can be done with that. Go to the next slide. Go to the next slide. Yeah. So what is this here? Can you read? Paracetamol. That is the chemical structure. Okay. Have you synthesized it in the lab? Yes. Okay. When you synthesized, you might have got mostly the thermodynamically stable polymorphic solid. Okay. If you use certain conditions, you can get a less stable one as well. There are two polymorphs for it. Paracetamol can crystallize in two different solid forms. One is the most stable one. Most of the paracetamol tablets have the stable form, but there is also unstable form which dissolves much better. It has all the other properties better, but stability is lower. So even tabletability, the way it gets compressed, is also much better for it, but this is not stable. If we can stabilize it using some methods, but still if we can get a good tablet, so this will be actually much preferred compared to the other one, because it has much better solubility and many interesting properties. So even a very simple molecule like paracetamol has multiple polymorphic forms. So this is very important for certain drugs where solubility is very poor. If you can change the solubility, improve the solubility, and you don't have to take lot of drug. If you can with very small amount of the drug, you will be able to make it more effective. Okay, next. So yeah. So have you studied this? BCS class 1, 2, 3, 4, right? Drugs are divided into 4 classes. So here 2 parameters are taken. One is the solubility, another is the permeability. Okay, based on that, drugs are actually divided into 4 classes. If drug has high solubility and high permeability is very good. You don't have to do much. You can directly compress and then you can take them into market. But if solubility is very low and permeability is also very low, most of the time you can discard that because it's very, very difficult to change this. But using crystallography knowledge, okay, if you can get a polymorph, or if you make a complex, or if you make a salt, okay, and then improve these two properties or at least one of the properties, then you can bring it to market. Okay, now a lot of people in this area from this solid state chemistry background are trying to basically take the old discarded drugs and then bring them back, for the administration. Okay, this can be done. Now a lot of research is going on. It's a very new field and still not many cases are in the market, but this is a very, very important area. So you can change all this by using crystallographic information. Go to the next slide. I think I am trying to finish now. So you can also form co-crystals. This is the last I am trying to conclude. So you can also, for example, if you have a drug, if you crystallize pure drug, you can get polymorphs. But if you take another molecule, for example, saccharin is a sweetener. If both of them form a complex in the solution and both together will crystallize and form a crystal. So that is called co-crystal. Okay, if they form an ionic complex, then you call that as a salt. Okay, you can make so many variety of solid state forms taking the same drug. So now you can make a library of these solid state forms and then one of them might show very good properties. Okay, so this area will allow you to actually modify the drugs. Even more difficult ones can be modified and then you can improve the solid state properties. So there are a lot of opportunities and scope if you know this subject. Go to the next slide. Yeah, so here one example here. So this is a theophylline. So one is anhydrous form and another is hydrate form. So as you can see, within a short time, so here dissolution is very good. So you have lot of drug in the fluids already, so absorption will be much better. So you can also see these are different forms. So if you see time wise, amount of dissolved drug in the solution, very different for different forms. So solid state modifications can give you lot of modification. So using that you will be able to actually modify the drug and then use it further. So next slide. So I think we can skip it. Next slide. Yeah, thank you very much. With this I conclude, and if you have any questions, maybe I'll be happy to take one or two questions. Otherwise we can close it. Any questions should we take or? Okay. Any questions or it's clear? Okay. So I think we will close it. Thank you so much.
Thank you very much, sir. Thank you very much, sir, for joining us. I request Dr. Professor Vilgar Sami, sir, to felicitate our chief guest.
Thank you so much, sir, for exploring the molecular crystals and the protein crystals for the advancement.
We have planned one career guidance by the one of the student who was in India, but now he has done at least 4 or 5 postdoctoral fellows, and he is working as a scientist in Doha, Qatar. And we would like to address the students about the pharmaceutical students' opportunities in the world. Shall we have it before lunch or after lunch? After lunch means you will sleep nicely. Okay, we'll break for lunch and we can have after the lunch. Huh? Okay, ma'am. And listen, please. Outside, it is very hot. So that's why many are not moving outside and sitting inside. You see, there are two counters, that is right side of the college for the girls. One counter is for the boys, for the MNR Pharmacy College students. For the delegates who came from outside college, staff members or students, we have arranged in the building first ground floor. In ground floor, 2 classrooms we have arranged. Understand? Our students wanted to give their classroom to other college students. Please understand, don't crowd, it is very hot. Unwantedly, don't make confusion and don't have friction. Again, I repeat, this side for girls, MNR Pharmacy College students, 2 counters, and this side is for boys, 1 counter, and other college students who came from different colleges, students and boys, that is delegates, in the ground floor, 2 classrooms, we have arranged lunch. Please join for the lunch. Thank you.